Now, we're not just talking about the metabolism aspects. We're going to talk about a lot of in our biochemistry series. We're going to be talking about fat metabolism. Now, we're not just talking about the metabolism aspects. We're going to talk about a lot of pathologies. We're going to talk about a lot of pharmac and everything related to fats. Okay, so let's get into it. Now, first things first, we're going to understand the structure of fatty acids, right? Now, acids, meaning they have a carboxyl group in the end. Now, they can be saturated versus unsaturated. Unsaturated meaning that they have double bonds in their chemical structure, right? Now, further classification, what you'll see is you have essential versus non-essential. Essential meaning your body doesn't make it, right? What are the two most important out of all these that you have to remember? It's either linoleic or linolenic, right? A lot of people get confused within it. Okay, what does it? What's the basic difference? They're both 18 carbon in length. The double bond placement is the one thing that differs between them, right? Of course, all the other amino acids that you see here are important because they make, you see, fatty acids and lipids. They're all making up that cell membrane, right? They all make up that the neuronal structures in the brain. So they're very, very important for our brain functioning, cellular functioning, everything, right? So it's important to remember how these structures sort of present with generally. Okay, so that's the basic introduction on fatty acids. Now, going into how you sort of take those fatty acids in from your food, right? Now, understand that triglycerides is two things. First is the fatty acids that you see here. I'm gonna zoom in further. These are the three fatty acids that you see with a glycerol backbone, right? That is why it's called a triglyceride, right? Otherwise, a lipid could have a cholesterol backbone. You see these four rings that you see here. This is a cholesterol backbone, right? Adding additional side chains to certain carbons makes it a cholesterol derivative, right? But those are the two things that you have to extract from your diet, right? Now, everything that is based on these two structures is what we're going to discuss, right? So first thing is, as you eat a fatty meal, you see lipase is the thing that actually cleaves the triglycerides, and it leaves what does it cleave? It cleaves two fatty acids off the chain, and then it leaves a monoglyceride or three fatty acids and just the glycerol backbone, right? Eventually you absorb all of this through the stomach. You make the gastric lipase and hydrolyze it, and eventually you go into the small intestine and absorb all of that, right? Now, couple of other things that happen with fatty acids, because fatty acids are not water soluble, you need to make them water soluble, right? In order to do that, you have to use your bile salts, right? Bile salts are hydrophobic on one end, hydrophilic on one end, allowing you to join these two, the fats in the food versus the water in the digestion tract together, right? And eventually you can get all these fat soluble vitamins and emulsify. That's the keyword. Emulsification happens because of bile salts. All right. Now, bile salts are recycled in the ileum, and what I need you to see is the word is called enterohepatic circulation, right? This whole reabsorption begins at the duodenum, but it ends at the terminal ileum, right? One of the main questions that they would like to ask is Crohn's disease affects the entire GI tract, but if it affects the terminal ileum, a lot of times you will see that the bile salts are going to be lost, right? A lot of fat malabsorption starts to happen in those kinds of diseases, right? So fat malabsorption due to intestinal breakdown and stuff like that, right? Those questions are related to this circulation, right? Or if there's any bugs and things like that, right? All of those are related here. Jejunum is best for short/medium chain fatty acids, right? Long chain fatty acids and plus monoglycerides uptake into the epithelium, reesterify to triglycerides. So what's essentially happening is you're taking triglycerides, you're breaking them down, you're taking them in, so it crosses that cell membrane, and once they're inside, you undergo reesterification, right? Make the triglycerides again, and then you package them into chylomicrons, and then you send them out into the bloodstream. Okay, a couple of important points that comes to just for triglycerides is this protein right here, MTP, microsomal triglyceride transfer protein, right? Basically, what these triglycerides are, how are they packed, right? This protein puts it into the apolipoproteins that the intestine has formed, right? And eventually these will go into the circulation, go up to the liver and all of that, but it's the MTP protein that's actually causing the packaging of these triglycerides, right? Apart from this, everything else that you see, we'll discuss it slowly, but that is just the introduction to triglyceride digestion and absorption. Okay, all right, moving forward. Now we're going to study the biosynthesis of fatty acids. Okay, now first thing is, we haven't gone through the glucose metabolism, but we understand the state in which fatty acids are going to be made within the body is the fed state, right? You have a lot of energy and you want to store that energy. Okay, so in that case, all those hormones, for example, if it's insulin or if it's norepinephrine, epinephrine, or let's say it's glucagon, etc., right? All those hormones have adjusted themselves so they understand that we're going to be storing these things. Okay, now once that pyruvate or glucose comes in, right? What you see here is the TCA cycle, right? TCA cycle is running, right? And what is the currency that states that you have high amounts of energy within the cell? That is citrate. Okay, as soon as the cell understands that I have high citrate molecules, citrate goes out of the mitochondria by the citrate shuttle. Here is there's two ways communication using citrate, right? In order to get citrate out, you have a singular transporter that gets the citrate out, right? But what happens if I need that citrate back in again, or I need a high energy signal back in again, right? In order to come back in, you use something called malate, right? So another name for these two things is called the citrate malate shuttle, right? This is how the internal mitochondrial membrane actually communicates with the cytoplasm in terms of energy currency. Now we got the citrate out. What ends up happening is you have to make it, in order to use it, you have to make it into acetyl-CoA, right? Now the main enzyme that you sort of use here, right, in order to get this done is this addition of this coenzyme A, right? The coenzyme A. Now we have to remember all these different things. I'm just going to show you the structure of coenzyme A. The reason I'm bringing this up is because we should know everything about these structures so we can relate it to other things in biochemistry, right? What we see here is this is the basic backbone, right? This is the adenosine diphosphate, right? It's the nucleotide paired with the sugar, right? What is the main ingredient that you're supposed to remember that forms the coenzyme A? Pantothenate, right? Pantothenic acid, okay? That is B5, right? So it's important to remember, these structures because they will come back eventually, right? So what you did was you took the citrate and you made acetyl coenzyme A, right? Now, acetyl coenzyme A undergoes a reaction called carboxylase. I need you to remember this right now. Whenever you see carboxylase being mentioned in any biochemistry reaction, it is called a step-up reaction. Why is it called step-up? You're converting a C2 molecule into a C3 molecule, right? Now, where's that C coming from? Where's that carbon coming from? It's coming from CO2, right? That's why the carboxylase. Carboxylase is called the ABC enzyme. That's how you're going to remember it. ABC, ATP, biotin, and CO2, right? So whenever you talk about carboxylase, ABC enzyme, right? And B7, biotin, is carboxylase dependent, right? So that's how we're going to remember all these now. Acetyl-CoA, step-up reaction, ABC enzyme, okay? Now, once you form malonyl-CoA, that's the base molecule, okay? That's the 3-carbon molecule that can go into fatty acid synthesis pathway, right? Now, so far, we've made the base molecule. Now we're going to elongate the chain, right? Fatty acid synthesis is done by a group of enzymes, right? And they have this very structured cyclic reactions, okay? First step, condensation, reduction, dehydration, and then reduction again. CRDR, okay? CRDR, okay? Condensation, right? When you condensate, what you see here is you're basically taking out a CO2, okay? So instead of this 3 plus 2 that you see here, in order to combine these, right, I have to take out a CO2 and make this 4-carbon molecule right now, okay? That is condensation. So expelling of the CO2, okay? Eventually, what you need to do is start reducing this. Now, when you reduce a ketone, you make an alcohol. And if you reduce an alcohol, you make that methyl group, right? So that's exactly what's happening. You see, I'm targeting this ketone, turning it, reducing it, making it an alcohol group. And eventually, I'm going to take this, dehydrate it. That means I'm going to take out the H2O, and then I'm going to make a methyl group right here. Okay. Now, eventually, what I need to also do is you have to dehydrate again, but this time you're going to use this energy, right? Very crucial, that you understand that the energy for fatty acid synthesis is NADPH, right? It's not NADH, it's NADPH, right? There's only a few locations and few reactions that produce NADPH, right? We'll go into that, but understand under energy, I'm taking the dehydration step and now I'm reducing again, right? Now, if you end up doing this over and over, you get this fatty acid synthesis train, right? You do it 6 times and you eventually end up with palmitate, which is a 16-carbon molecule. Okay. Now, once you've made it, you're good, right? You have, a basic structure, right? Maybe elongate it to 18 or even 20, right? You can undergo, unsaturating the whole fatty acid to make, specific types of fatty acids and stuff. And they've given you a little bit of detail about that, but you don't actually need it apart from remembering a couple of different things, right? Desaturation might take place in the mitochondria. And in order to do that, they have to go back to the, mitochondria. And we'll talk about how they go back into the mitochondria, right? And once you do that, you may have this enzyme, fatty acyl-CoA desaturase, making that unsaturated fatty acid chain, okay? So that's the entire, formation of these fatty acids, okay? All right, so just to refer back to the first aid, right? Here is the synthesis pathway. They've given you all these things, for example, insulin versus glucagon controlled by ACC, and then eventually you make all the steps. We discussed a lot within just this one step, right? But it's important to know all these because they will ask you questions about it, okay? All right, now, since we've created it, we should also know how to destroy it, right? Because that's essentially how you're going to get energy out of these molecules, right, when you need them. So in order to, destroy these, destruction happens within mitochondria, right? And we also saw unsaturation of the fatty acids also takes place within the mitochondria, okay? Now, how do these fatty acids go inside the mitochondria? They're through the carnitine transporter, right? Carnitine is a protein, right? So you'll see one carboxyl group, you may see an ammonia group, and then you have this hydroxyl group attached to it as well, right? It has everything, right? Now this is a quaternary um, nitrogen compound, right? So that means it's a very, very electropositive, right? Now, because of this, what ends up happening is you go into the actual system, right? You take the fatty acid, right? You combine it and make it into fatty acyl CoA, right? You use that coenzyme A again, right? Now, once you combine it with energy, you have to take this coenzyme A and attach it to the carnitine, right? So it attaches to itself right somewhere around here, right? You see the hydroxyl group has made this group right here, right? Now, once you attach it, then this acyl transferase will take it on, on the inside, okay? Once inside, you see there's another carnitine transferase, but in this case, what it does is it takes that carnitine, separates out the carnitine and recycles it, so it can be used for transfer again, right? And it also separates that fatty acyl CoA and the coenzyme. Okay, so you separate the carnitine and you take out the fatty acyl CoA, and eventually you go into the beta oxidation. Okay, so I hope you understand that the carnitine shuttle is just between the outer and inner membrane. Doesn't have to do anything with the reaction of the beta oxidation, right? And it is an amino acid that is required by the CAT1 and CAT2. Okay, now exactly what we did for biosynthesis of fatty acids, we're going to try to reverse that and make the chain smaller. Okay, you remember I said CRDR, condensation, reduction, dehydration, reduction, right? So what we're going to do is we're going to reverse these, right? So the steps are oxidation, hydration, oxidation, and then release. Okay, so you see, when you oxidize, you lead to, first of all, formation of a F8DH2, and then you get the NADH out, right? Now, eventually, you take that and release the 2 carbon, so you get a 2 carbon molecule and you get a shorter fatty acid, okay? So if you go from 18, now you're coming back to 16, right? From 16 to 14, and then you keep going until either you reach an odd number or you have finished the entire chain altogether, right? Let's say if it's just an even number, for example 20, 22, or 24, it's going to finish everything in that much, that many cycles, and all you have is like 5 acetyl CoA or whatever, right? But if it's an odd number chain, what's going to happen is it actually leads to that acetyl CoA combining with, like, let's say an odd chain fatty acid, right? Combines it and makes this methylmalonyl CoA, right? Why does it do that? Because during carboxylase, it ends up adding, right? It ends up adding a CO2 in it, right? Now, if you make that methylmalonyl CoA, you can, mutate it. You can go into the succinyl CoA direction, or you can also use it for gluconeogenesis. Okay, but this is slightly different, right? We'll see it when we do gluconeogenesis. I'll mention methylmalonyl CoA and all of that. But yes, this can go separately into like gluconeogenesis. Doesn't have to come back to acetyl CoA, structure. Okay, so that's what's happening, right? Odd number chain fatty acids, they have to undergo this intermediate step into formation of methylmalonyl CoA. Okay, I hope this makes sense with the carboxylase coming through, right? Eventually, all of these will go into the citric acid cycle, and then all the contents of that will go into the electron transport chain. Okay, all right. So now we're going to discuss all the fatty acid metabolism problems. Okay, now what you see here is whatever the process we've discussed, something is wrong in that process. Okay, so first of all, the first thing that they will talk about is just carnitine deficiency. Okay, you don't have the proper amino acid formation, or let's say there's some internal metabolism error, you're not making that carnitine. Okay, or maybe you're not absorbing that carnitine. Okay, what ends up happening is all those long chain fatty acids, right, they cannot enter the mitochondria anymore, so they cannot undergo breakdown. So what do they end up doing? They start to accumulate in the cytosol, right, eventually becoming toxic, eventually leading to degradation. Right, so any system, right, who are the main organs that handle fatty acid breakdown? First of all, it's the liver, because liver is going to generate the energy when you are fasting and stuff, right? Cardiac tissue, skeletal muscle tissue, right? Those muscles will not receive any energy after a long time, right? And they will start to undergo degradation. So we understand systemic primary carnitine deficiency. Now, next comes medium chain acetyl CoA dehydrogenase. Okay. Now, what does this mean? It just means that fatty acids are divided into 3 categories. There's very long chain fatty acids, there's long chain, and then there's medium. Okay, medium and long chain are handled by the mitochondria, but very long chain fatty acids are handled by the peroxisome. Okay, and I'm going to go into the peroxisomal metabolism, right? But in this case, in MCAT deficiency, what you see here is break down these medium chains anymore, right? The fatty acids, again, they start to accumulate, and they damage, right? So it's just about the length of it. For example, let's say it's 8 carbon long, 10 carbon long, 12 carbon long, right? They're not going to be able to be degraded, and you have less energy in those tissues, and eventually they accumulate in the cytosol. That's it. Okay. Next comes CPT2 deficiency. Okay. Low CPT2 activity again leads to acyl carnitines accumulating within the skeletal muscles and stuff. Now the difference is acyl carnitines. Here it was fatty acids. Here it is a carnitine, right? Because CPT1 is working fine, is binding the carnitine, but there's no carnitine dissociation. Okay, so you see that's why you have long chain acyl carnitines, and you have total and free carnitine decreasing, and also a lot of fatty acids being consumed and not making enough energy in those tissues. Okay, what do those tissues do? They die, right? They eventually start to die. Okay, now common diagnostic features that they've given you here: weakness and hypotonia. Of course, why? Because you don't have energy. Your muscles are gonna start to break down. Liver dysfunction, because even the liver cells need energy, right? So hepatic encephalopathy, hyperammonemia, and all that, because your liver is not doing its job anymore, right? It's damaging and it's accumulating ammonia, and it's damaging your brain, crossing the blood-brain barrier, etc. Okay. One of the most important things that they will talk to you about is hypoketotic hypoglycemia, right? Ketones are a byproduct of fatty acid metabolism, right? Fatty acid breakdown. Okay. Now hypoketotic hypoglycemia is happening because there is no fatty acid breakdown, right? So if a person is coming to you with hypoglycemic symptoms, you measure their ketones, or you measure the ketones in the urine, and there's nothing, you're concerned, right? Why isn't this person making ketones? Because you're supposed to. That's just physiology. So that's when your eyes should open up, and you should think about something like fatty acid metabolism problem. Okay. Now, because all of these fatty acids, they might spill over, they're also degrading the structures and stuff, you will start to see lactic acidosis, just plain old metabolic acidosis. Okay. Now, because you see primary carnitine and CPT2 deficiencies, you see massive amounts of cell breakdown, muscle breakdown, right? And even cardiac tissue breakdown. Okay. And what is the management of all of this? It's better to go through the first aid for these points in order to differentiate them. I'll open it up. But the management of all of these, right? Systemic primary carnitine deficiency. Don't let them fast, because that's what beta oxidation. Within the fasting state, you're gonna have most amount of beta oxidation. And within the acute setting, give them carnitine and give them dextrose, right? Carnitine will help. If you supply that deficient person with carnitine, it's gonna help them, right? And give them dextrose so they slowly go from the fasting state to the fed state. Okay. Now look at MCAT deficiency. What you should do for these guys, again, avoid fasting and give them diet, which is rid of these medium chain fatty acids in general. Okay, you give them high carb diet, or even in acute setting, give them IV dextrose. Okay, CP2 deficiency, avoid fasting again. Here you increase the amount of medium chain fatty acids, right? And what that should sort of do is, and even the carbs, and that should take the burden off of that CPT enzyme, and allow the mitochondria to rest a bit. Okay, so just so we understand it, here's the thing again with systemic primary carnitine deficiency, LCFAs, dilated cardiomyopathy, and I hope all of this makes sense now. And then MCAT deficiency, you see hypoketotic hypoglycemia again, liver dysfunction, hyperammonemia, the sudden death in children, because it's very hard to have the patient survive, especially a child who needs constant nutrition, and it's even the milk has a lot of fatty acids, right? And they need to process that. So it's a very you may see it early on. Okay. Now, before I go into the very long chain fatty acids, I want to finish up this ketone discussion. Okay. Now, there's two types of ketones within the body. It's important to remember them. Okay. So first ketone that you're going to see is beta hydroxybutyrate, right? And the next one is acetoacetate. Which one is the more stable one? It's beta hydroxybutyrate. Okay. And that's the most abundant one as well compared to acetoacetate, right? But they function back and forth, right? So these are the two ketones. It's important to remember their name because they're options, right? In certain questions, they will come up as options. Okay. Now, in the liver, fatty acids and amino acids are metabolized into these two things, right? In prolonged starvation, you see ketoacidosis occurs, and oxaloacetate is depleted for gluconeogenesis, etc. Right? It spills into the blood, right? Eventually, it may turn into acetone, and it can be expired by lungs, so you get that fruity smell in their breath, right? Or it's gone into the skeletal muscles, and that can also convert it back into some sort of energy as well. Okay, so that's the main reason why ketones are spilled over, right? They're still energy capable. They can still deliver you some energy, but what it ends up actually doing is causing a lot of metabolic acidosis. Now, the urine test that they're actually talking about is called the nitroprusside test for ketones. Okay, what does it actually measure? It's acetoacetate, not beta hydroxybutyrate. Okay, so if acetoacetate is there, you put the the reaction reagent in here. Eventually, you're gonna have just a color scheme telling you how many ketones you have, right? If it's very dark, they're gonna say ketones 3+ or something, right? That's how the nitroprusside test works. So that's what they're talking about over here. Okay, RBC. Now this is important, right? Who can actually use ketones versus who cannot? So as you see, liver cells lack beta ketoacyl-CoA transferase, so they cannot use ketones for themselves. So the liver cannot use it, the RBCs cannot use it. Every other organ though they can take that ketones and convert it into some sort of energy, as you see here. Okay, so what we see here is they've compared 3 different pathologies, right? Now I'm going to go through all these eventually, but right now we're just going to focus on the MCAT deficiency that we need, right? You see ketone levels being low, glucose levels being low, right? When we do gluconeogenesis, these 2 will make sense, but we sort of understand it because we've seen it, right? Methyl methanyl CoA, right? Propanol CoA. All of those were gluconeogenic enzymes, right? One of the ways that you saw that was odd chain fatty acids, right? You see ketone levels can increase when you're in gluconeogenic problem, but glucose levels will be low because you're not actually generating glucose, right? You can only use ketones for your energy in that case. Okay. Next thing we should take a look at is a lot of questions they will pose to you is when did the person start to deteriorate post fasting, right? Your energy stores are not built the same way, right? Within 2 seconds you're going to consume your ATP. Within 10 seconds you're going to consume your creatine phosphate. Within a minute you're going to consume you're going to start to make the anaerobic metabolism, right? Eventually, after decent amount of working out, you start to first use your glycogen stores. Once you get rid of those glycogen stores, then you switch over to fatty acids, right? A lot of people think going to the gym and working out, you're burning fat right away. No, after a decent amount of time, after at least a couple of hours, do you actually consume all your muscle glycogen, and do you actually go into actual fatty acid breakdown, right? And eventually, even when you're post-workout, you're recovering, that's when you actually you're using your fatty acids to make energy, okay, and replenish your glycogen stores, right? So I hope that makes sense, right? Here's the detail on it, and you see when you start to use the fat versus the protein, because eventually, let's say someone's actually undergoing malnutrition or something, right? At what time is the adipose tissue going to be finished, and at what time are you going to start to eat up your protein, right? So that's what they're talking about here. Okay. Now you see it's mentioned here, peroxisomes, beta oxidation of VLCFAs, strictly peroxisomal process. There's a thing called alpha oxidation, right? The difference between alpha and beta is just which carbon does it actually break down. If it's right next to the COH, then it's alpha. If it's right next to that alpha molecule, then it's the beta. Okay. So alpha oxidation is also taking place within the peroxisomes. Catabolism of amino acids and ethanol as well can happen in the peroxisomes. Synthesis of bile acids and even plasmalogens, right? All of these are parts of the brain, they form the brain and stuff. All of them are being taken place in the peroxisome, okay? So it's very important to remember, how important peroxisome is, right? Just a quick summary of all these 3, and then we'll go into depth, right? Zellweger is a mutation in the PEX gene, right? PEX means you're not able to make the peroxisomes properly, right? What ends up happening is, because you cannot degrade any of these things, very long chain, branch chain fatty acids, amino acids, all of these things accumulate within the peroxisome and eventually lead to death of the cell. Okay, so that is Zellweger. Refsum is a problem with alpha oxidation. See, what's happening is phytanic acid, which is a plant-based acid, right, fatty acid, it's not being degraded and it ends up accumulating within all these different cells, right? Causes joint problems, skin problems, eye problems, right? That is Refsum. Adrenoleukodystrophy is a problem with beta oxidation of VLCFAs. Okay, why is it happening? ABCD1, right? Adrenoleukodystrophy something, right? But ABCD1, right, leads to adrenal problems. Leuco, meaning the white matter in the brain, right? Those 2 are the main things that are undergoing problems in this case, right? Of course, you might see testicular dysfunction, but that's the main essence of these. Adrenal gland and white matter of the brain is being destroyed, right? But now, if you have the summary, now we can go in one by one, right? First thing is looking at this picture right here. This is the Zellweger syndrome patient, right? So you see seizures, craniofacial abnormalities, right? Loss of vision, and eventually you'll see hepatomegaly, and all of this is autism or recessive. Okay, that is Zellweger, right? Refsum. Now, Refsum, as I said, you see the image right here. Phytanic acid is supposed to be broken down and it's supposed to be used by the mitochondria for energy. But here you don't have the enzyme working, and eventually it all accumulates and it leads to toxicity within the peroxisome, right? What does it sort of look like? You see, accumulates in the retina, causes retinitis pigmentosa. Accumulates in the nervous system, leads to peripheral neuropathy, right? Accumulates in the skin, leads to ichthyosis, which basically means skin, like snake skin, like, okay? Ichthyosis, which is like a dry patch in that area, okay? Accumulates in the bones, leads to dysplasia within that bone, okay? Very, very specific symptoms you're seeing here, right? That is Refsum disease. Skin, bone, neuron, and eye, okay? That is Refsum. Then we go into adrenoleukodystrophy. Okay. The thing is, in many questions, they like to confuse you with several types of leukodystrophies. Okay. There's 3 main that they love to ask about all the time, right? But first, we're going to learn about adrenoleukodystrophy. Okay. We know all of this. We've seen, the pathophysiology, but understand the clinical features. Okay, you see this is degeneration that you see here. This white entire thing, right? It's all degeneration. It's supposed to be nice and black right here, right? But all of this is degeneration. Degeneration, mostly in the occipital region, right, and even in the brainstem region, is seen with adrenoleukodystrophy. In addition to that, you see adrenal cortex and even the Leydig cells in the testes, right? So if a child or like a young teenager or someone comes to you with these types of symptoms, right? Sexual problem, right? Sexual maturity problem, maybe adrenal cortex problem, which means like low BP, adrenal insufficiency like symptoms, right? And then they come to you with neuronal problems like maybe seizure or maybe trouble coordination. Now what do you see? You see there's a couple of treatments that they might use, but they're not very proven, right? So macrophages contain long chain fatty acids. Treatment is something called Lorenzo's oil, which is basically triglyceride mixture. They're trying to make sure you're not consuming very long chain fatty acids too much, right? Because that's the problem. That's the source of the problem, right? And they might manage it through that mechanism. Okay, right. So don't get confused. This is the thing that is called X-linked adrenoleukodystrophy. Okay, this is the thing, right? X-linked. So it's X-linked recessive, right? When we do genetics, I'll make sure you understand everything about that. But yeah, okay. Now next comes metachromatic leukodystrophy. You see, this is a lysosomal storage disease. Okay, and even the next one is a lysosomal storage disease. But you have to understand it here because it's a leukodystrophy, right? Again, white matter of the brain is going to be affected. What's happening here is you're accumulating something called sulfatides, and when we do LSDs and stuff like that, you'll understand it better. But right now, there's these things called sulfatides, right? They're accumulating within the CNS cells, and they're eventually, going to lead to ataxia, dementia, etc. Right? Even in young children, juvenile, or even in the adult forms, you'll see multiple, areas of demyelination happening, right? And what's the sort of CT scan differentiation you see? Metachromatic leukodystrophy affects all different types of parts, right, of that, white matter, right? Surrounding all these different ventricles, you're going to see metachromatic leukodystrophy, whereas adrenoleukodystrophy is sort of self-limited in presentation. Okay, hope that makes sense. Now, last one is called Krabs disease. Again, this is also a lysosomal storage disease, right? It's also called globoid cell leukodystrophy, and what you are accumulating here is galactocerebrosidase, right? That's the deficiency. But galactocerebrosides are accumulating. They not only accumulate within the CNS, they accumulate within the peripheral nervous system too. Okay, so peripheral neuropathy, optic atrophy, and just developmental delay in general. Okay. And if you look at the presentation, it doesn't have that specific sort of spread like the other two, but it's definitely located within that central brainstem / ventricle region. Okay, so they might not give you a CT scan for this because they're very hard to distinguish findings, right? But we have to understand where the relation between the three. Okay, they're all causing demyelination and destruction of the CNS and PNS. Okay, so that is about leukodystrophy. All right, guys. So now what you see here is the lipoprotein structure, right? What are they used for? It's for carrying lipids and triglycerides throughout the plasma, right? Because they cannot dissolve in, and if they're there in the open, they might activate enzymes, etc., right? So they carry fats. Now, in order to carry fats, what you need to do is you need to have certain IDs, right? And IDs are called apolipoproteins, right? Apolipoproteins help in identification of where this lipoprotein is coming from and where it might be going and what functions it may be performing, right? That is the specialized way of looking at it, right? So you see this is the structure. You see it has this membrane again, and within that membrane you see these embedded proteins, maybe an integral protein, maybe a peripheral protein, basically denoting where is this made and where is this going to go. Okay, now if you understand that, the best way to go about learning the functions and the markers and all that is by just going through the cycle itself. Okay, so we start here, right? We figured out how triglycerides are taken in, right? The MTP protein, putting it into what is called the chylomicron, right? Now, chylomicrons have a special type of marker, right? The first marker we have to discuss is the B48. Okay, B48. You see, apolipoproteins are just amino acids, right? A link of amino acids. A quick question that they like to ask you is, you see the difference between B48 that you see here and then B100. It's the same sort of gene. One is transcribed fully, and the other one is halfway transcribed. Okay, so that's the difference between the two. So B48 is half transcribed, is up to 48% of the length, and B100 is 100% transcribed, right? B48 is a special marker for intestinal origin. Okay, if something is made in the intestines, it's going to have B48 in it. Okay, now what you see here is B48, right, has that, all the triglycerides in it, as well as C2 and apolipoprotein E. Okay, as soon as this chylomicron goes into an adipose tissue or a muscle tissue to, deliver that free fatty acid, it loses something in this, journey, right? It's the C2. Okay, C2 is what carries out the function. Okay, I hope that makes sense, right? Now, just so we can revise it, this is on page 111, and this is what we're sort of discussing right now, right? So you see B48 mediates chylomicrons, secretion into the lymphatics, and then we saw C2. This is called lipoprotein lipase cofactor. Okay, just so we understand what lipoprotein lipase means, lipase, right? Degradation of lipoproteins. Okay, so it takes up all the triglycerides, breaks it down, and then stores it in the adipose tissue. Simple. Okay. Last one is ApoE, mediates remnant uptake. So if anything is going back to the liver, it will allow the liver will reuptake it if it has ApoE on it. That's how simple it is. Okay, so origin versus function versus target, right? Or the end of that structure. Okay. Now, as we move forward, now we're going to talk about the VLDL and all of that. Okay. Now, VLDLs are made by the liver. The liver takes up all the cholesterol, it takes up all the triglycerides, packs it into the VLDL. Right. So if the liver is making it, it's the B100. Now, again, C2 comes in because we need to unload that triglyceride. So C2 unloads it and gives it to the adipose tissue, muscle, or wherever triglycerides are required. Right. Now, in while doing so, VLDL is eventually converted into IDLs. Okay, intermediate density, where you see both the cholesterol level and triglyceride level is the same. Right. Eventually, what can happen is you may see hepatic lipase. Right. And what do we know about hepatic lipase or hepatic structures in general? They're dependent on ApoE. Right. So after the reaction of hepatic lipase, under the direction of ApoE, all the triglycerides are removed, and you end up with LDL, which is just mainly cholesterols. Right. Now, LDL has only that one marker left, denoting that liver origin. Okay. So how do we know where does it end up? Right. The thing is, LDL doesn't need an apolipoprotein for its the end of its journey. Right. It doesn't need a marker for that because it has its own receptor. Right. Every tissue that is capable of undertaking LDL, right, has its own receptor, LDL receptor. Right. LDL receptors specifically that are part of the liver take up the LDL receptor, and they use it for their cholesterol synthesis, etc., hormone synthesis, whatever. Right. So if for example, you will study about congenital adrenal hyperplasias, right? How is cortisol made? How is the aldosterone and all of that made? Right. They're all cholesterol derived hormones, right? Testosterone, estrogen, all of those are cholesterol derived. So all those tissues have their unique LDL receptors, and they receive the cholesterol from LDL. Okay. Now, once the LDL is delivered, what you end up seeing is you see extrahepatic tissue taking up the cholesterol. There is a molecule called HDL, right? HDL is usually made by extrahepatic tissue, so it doesn't have any liver origin markers, but it has two unique markers, A1 and A2. Now, let's look at A1. Found only on alpha lipoproteins, which is HDL, activates LCAT. Okay, what is LCAT? LCAT is the enzyme which basically takes cholesterol from lipoproteins, right? Let's say LDL, or you can take it from tissues, right? So LCAT is the enzyme that loads cholesterol back into the HDL, so it can go back to the liver, right? So you know how they say LDL is bad cholesterol, HDL is good cholesterol? This is the reason why, right? Because LDLs deliver to the tissue, HDLs take it away from the tissue, right? And then we can just sort of go through all these enzymes that are required, right? So CETP, right? Mediates transfers of cholesterol esters to other lipoprotein particles, right? So if the HDL decides I want to give this cholesterol to VLDL, IDL, or LDL, can do that cross-functional transfer via CETP. Okay, hepatic lipase, we know what it does. It degrades triglycerides. Hormone-sensitive lipase, we know what it does. It degrades triglycerides under different conditions, right? We just understood LCAT. We know what lipoprotein lipase does, right? Takes out the triglycerides in through to the adipose tissue, right? Via chylomicrons or even VLDL, right? You also have pancreatic lipase used for digesting and absorption of triglycerides. And then the last one is this PCSK9. Okay, it's important to recall this here and now. Okay. PCSK9 is a enzyme that degrades LDL receptor, right? So if you come up with a drug that inhibits PCSK9, right, that inhibits this enzyme, what have you essentially done? If you inhibit PCSK9, that means there's going to be more LDL receptors located within the liver now, which means you're going to end up taking a lot more of those LDL molecules back to the liver and not allowing those LDLs to deposit within the tissue. Okay, so I have just explained the drug to you, right? So I hope PCSK9 makes sense, right? It's naturally supposed to degrade the LDL receptor, but what you see in, if you inhibit it, you're, increasing the recycling. You're gonna lower serum LDL because all of it is gonna go back into the liver. Okay. Now, what we're gonna do is we're gonna go into the drugs, right? All the hyperlipidemia drugs. Okay, so let's go there first. On page 345, all the lipid lowering drugs are there. We've seen how they work already, right? All we need to figure out is the pathophysiology and the adverse effects, right? First thing is statins. Okay, statins, they lower the LDL quantity, right, by inhibiting the hepatic synthesis, right here, hepatic synthesis of cholesterol, right? Because they inhibit the hepatic synthesis, they also lead to some amounts of liver damage, right? Because they inhibit hepatic synthesis and even cholesterol synthesis in other parts of the body, they might lead to degradation. For example, a lot of muscle aches happen because of it, right? I read somewhere they also inhibit the coenzyme A formation, right, which is also a very high, a highly important energy in energy metabolism, right? So that's why the myalgias, hepatotoxicity, etc. Right? So that is how statins are working, right? You decrease the overall cholesterol, you don't have anything circulating, it's good for the person, right? It also has a lot of secondary effects. It's not just this, it reduces inflammation, right? It reduces the mortality rates, and as you see in coronary artery disease patient, because it decreases the overall plaque instability, right? So it helps in a lot of these secondary processes. It's not just the cholesterol that you're dealing with. Okay. Now, as we go down, let's look at all of these that are over here. Okay. First thing is, let's say I'm having a lot of cholesterol in my diet, right? What I can do is I can use these two categories of drugs. One is called bile acid reabsorption, right? So what these guys do is these guys let you waste your bile acids, so you never emulsify your fats and you never take your fats inside the intestines. Okay, these are called cholestyramine, cholestyramine, cholestyramine. Well, you would think, what would they end up doing? You see, all of those fatty acids and even the bile is coming out through and going into your intestines, even your large intestine, right? So it's going to create a lot of diarrhea. It's going to create a lot of GI problems, right? Eventually, if you use it and you're not supplementing yourself, you might even see fat vitamin deficiency, right? ADEK deficiency, right? So that's the adverse effects of bile acid reabsorption. Need you to remember the name. They all have the chol. Chol is the base word, chol, right? So bile acid reabsorption. Cholesterol absorption, right? If you're just focusing on cholesterol not getting through and going into your intestinal cells, ezetimibe, okay? Ezetimibe is just focused on that. So your overall LDL will decrease, okay? So here is LDL decreasing, and then you have LDL decreasing here. This might increase a little bit of HDLs, right? But the main thing being GI upset in both of these, okay? This can also lead to LFTs increasing because you're not essentially giving your cholesterol to your liver to work with, right? So liver might have some sort of damage, or there might be a secondary phenomenon happening too. All right, next comes fibrates, okay? Fibrates are a part of the lipoprotein lipase inhibitors, right? And we just saw what lipoprotein lipase was doing. It was inhibiting the overall, it was assisting in unloading of triglyceride, right? So if I upregulate the lipoprotein lipase, that means the entire triglyceride will go into my fatty tissue, right? It won't circulate, which is a good thing. I don't want it to circulate, because if it circulates, it causes hypertriglyceridemia. It can accumulate, it can lead to pancreatitis, etc., right? But if I give someone fibrates, I can decrease the blood level of triglyceride and store it into tissues, right? The way it does that is that it goes inside the nucleus and it activates this factor, right? PPAR alpha, right? PPAR alpha also induces HDL synthesis, as you see here, right? And just for a clinical correlate, there's a thing called PPAR gamma, right? That's activated by like the thiazolidinediones, okay, like glitazones and stuff. So just so we make that correlation, you see PPAR gamma over here, okay? Even this sort of deals with fatty acid storage and glucose metabolism, right? Both of those are just cousins in the same family, and what they end up doing is they sequester fatty acids. They lead to fatty tissue storage becoming more and more, right? So weight gain is a very common thing. Okay, you can definitely find a diagram that helps you elucidate a bit more, but that's more than enough needed for the exam. Okay. So we understood how fibrates are working. Now, a couple more, niacin, right? Niacin helps in a lot of HDL formation. The problem is what you can lead to is if you give them too early, you will end up making a lot of prostaglandins, right? They have flushed faces, right? And also you may see hyperglycemia happening and hyperuricemia happening. So not for diabetics and definitely not for gout people, right? So those contraindications are very, very important when it comes to niacins, right? PCSK9 inhibitors. Now you have a name to that. Alirocumab, evolocumab, right? Ocumab, right? That's the base word. So you're inhibiting the LDL recycling. So you're increasing the LDL recycling, and it leads to LDL clearance and removal from the bloodstream. Okay, leads to maybe myalgia, some delirium, dementia. You see, these are the only ones that are leading to neuronal symptoms. So you can't give it to a neurodegenerative person. Does that make sense? Right. So I know the adverse effects are written, but think about the pathologies that are related to those contraindications. Right. And of course, giving healthier fatty acids will decrease the overall levels in general. Okay, so go through this, make sure you understand all of this now. Okay. Now, next we're going to go into dyslipidemias, right? Naturally, just imbalanced fatty metabolism, right? So what you see here is lipoprotein functions and all those things that were in here. We went through all of them. A beta lipoproteinemia. That's the first thing we're going to talk about, right? Now, if you remember, I brought this up in the beginning of the session. MTP, right? MTP packs triglycerides into the chylomicrons and even VLDLs and stuff, right? If that is absent, you're not going to make anything. Eventually, you'll see APO B48 and B100 are lost. They're not there. What ends up happening is you're not absorbing any fats, right? So fat malabsorption, steatorrhea, failure to thrive. Your cells are not getting this crucial molecule, right? Eventually, everything will start to undergo degeneration, starting from the retina, starting from the brain, parts of the brain, and eventually, because you don't have vitamin E, you might see more and more things, right? Vitamin E deficiency needs to be learned because vitamin E is responsible for a lot of like cellular metabolism for fats. So that is about it for our discussion about everything related to lipids and fat metabolism. Okay. Let me know if you have any doubts, and I'll try to answer them right away. Okay. And affects all these different, cell protection, cell functioning and stuff, right? So all of these are layered sort of presentation. It's not just the loss of, lipids, it's the loss of vitamin E in general. Okay. So do review vitamin E deficiency from the earlier sections. Okay. Now, what you see in the biopsy, you may see that the intestinal cells are filled with these lipid droplets, but they're not being used. So they're just, nice and clear, but they're not actually doing anything, right? So they're just sitting there. So lipid-laden enterocytes. What are you going to do with them? You're going to give them, you're going to restrict long-chain fatty acids, and you're going to give them vitamin E. Okay, so that is A beta lipoproteinemia. It's in the name, right? Beta. So the B proteins are lost. Okay, now let's go down all the different familial dyslipidemias. Okay, hyperchylomicronemia. Right? What's happening? You have high amounts of chylomicrons, which means you have high amounts of triglycerides. Right? What is sort of happening here? Lipoprotein lipase can be deficient, or ApoC2 is deficient, right? And both of them are doing the same thing. They're trying to unload triglycerides, right? What is it leading to? Pancreatitis. That's the main thing. Acute pancreatitis, right? Or maybe even chronic pancreatitis. Okay. Why is that? Because triglycerides, they're droplets, right? They stick together. If they end up sticking together, they end up blocking tubes. One of the main being pancreatic duct. If you block the pancreatic duct, all the enzymes that the pancreas secrete in order to digest food digest the pancreas. Trypsinogen in general, right? So trypsin ends up degrading the pancreas and causes pancreatitis. Okay. Now hepatosplenomegaly can also be there, right? And you will see maybe xanthomas being present in the skin, right? Because you don't have that much effect of cholesterol, you may not see high risk of atherosclerosis, right? But triglycerides being high is a problem on its own. Okay. Now, a lab sort of feature you may see is they may say creamy layer in the supernatant, right? When you spin the blood, you may see a lot of that whitish creamy layer within that blood. Why? Because there's so much triglycerides in there. Okay. Next comes hypercholesterolemia, right? Absent or defective LDL receptors, right? So you're making a lot of LDL receptors, but you're not capturing them back, right? And what was the marker that was helping you understand where the LDL is coming from? ApoB100, right? So what is happening in this? High amounts of cholesterol in the system, right? High amounts of cholesterol. So of course, what's going to end up happening is you're going to get absurd numbers of cholesterol, and you're going to get accelerated atherosclerosis, right? So young person coming with MI symptoms, what is the problem, right? Why are they doing this? Like, what is going on, right? The question needs to be answered. You may see hypercholesterolemia. I think this one is generally the most common out of all these four, at least. Next comes dys-beta-lipoproteinemia. You see, this is A beta lipoprotein. This is lipoprotein, right? So this is ApoE. Okay. Now, if you remember, ApoE was supposed to capture chylomicrons and VLDL and take it back to the liver. If it doesn't, you see high triglycerides and high cholesterol in this case as well. Okay. So you get the xanthomas, but you also get the atherosclerosis. Lastly, just hepatic overproduction of VLDL leads to hypertriglyceridemia, absurd amounts, right? Acute pancreatitis in this case.